Hello. Today we're going to talk about how to quantify pathogens easily using Accugen's technology. Absolute quantification is the gold standard for quantifying pathogens. If you perform a quick PubMed search, you can easily find multiple articles referencing this method for virus, bacteria, and parasite research. Even though the technique is widely used, the most commonly faced problem in the lab is to generate a separate standard curve for each gene of interest. So if you're analyzing multiple genes, then it can be very time-consuming and cost reagents. Another less discussed problem is that the standard curve amplification efficiencies may be different to the gene of interest and produce inaccurate estimates. Therefore, we aim to solve these problems by having a universal external calibrator that can measure multiple amplicon lengths and is not influenced by the amplification efficiencies. Accugen's product consists of Accugen, which are the DNA standards, and RealCount, which is a complementary software. Accugen itself is a 90 base pair proprietary double-stranded DNA, and it helps the RealCount software in generating a standard curve that looks like this. On the right, you can see the process of pipetting Accugen standards, marked in red. Blue and green are your gene of interest, and essentially multiple sets of genes can be quickly measured on the same plate without making new standard curves. This tool is compatible for amplicons between 60 to 500 base pairs. As previously discussed, a potential problem with absolute quantification is that amplification efficiencies for the standards may vary to the efficiencies of the gene of interest, and that would lead to inaccurate quantification of your DNA copy numbers. We've designed Accugen so it does not need to rely on amplification efficiencies. Here you can see Accugen fluorescence remain constant in the PCR reaction. Let's have a look at the RealCount software interface. After qPCR has been performed from your machines, the raw fluorescence data can be exported and imported into RealCount, like this. Bottom left is the standard curves as you have seen previously. The left middle section shows the amplification of your genes. The software uses this to calculate efficiencies from the window of linearity. And the top and the right is where you can configure the settings. A more detailed tutorial can be found online for configuring these. In the configuration, one of the important variables is to enter the base pair length. This allows the software to calculate the theoretical copy numbers based on the fluorescence intensity per base pair. On the operational side, you just need to press Analyze here. And instantly, your results will show up as copy numbers here. Let's take a look at how the software does its calculation. RealCount generates a standard curve by plotting the raw Accugen fluorescence against the weight in nanograms. The fluorescence for your gene of interest, for example 12.5, is measured against the standard curve, obtaining amounts in nanogram. Once this is obtained, in combination with your previous amplicon base pair input, it back calculates to a set of theoretical copy numbers. One advantage of this software is that we had adopted a concept based on a paper published by Reuters in 2009. In this paper, Reuters discussed about how multiple software vendors had produced inaccurate results and should measure the amplification efficiencies from a window of linearity. If you use Accugen's real-count software, you're guaranteed to use the adjusted window of linearity algorithm and produce a more accurate and less biased result. Here in this slide, we will discuss about Accugen's consistency. We aim to design a method that can reduce discrepancies between different labs. So we have tested against a range of machines and diets from independent laboratories. In the trial, we gave the labs Accugen and lambda DNA of known concentrations. You can see here the results are fairly similar between different machines and master mixes. At the bottom is labeled as machines. In here, the lambda DNA had produced the expected copy numbers. Averaging those produced a line of best fit that has a fairly nice slope of the averages, as you can see. Let's look at an example of a case study. The experiment was to examine the level of IL-7 receptors on activated human PBMCs. As you can see, the control had high copy numbers, and the activated cells had reduced IL-7 receptor copy numbers. The results was produced quickly by real-count software, and you can generate results quickly like above. More details of the method and examples can be found on a paper on BMC biotechnology. In conclusion, you can quantify pathogens easily via Accugen and real-count. It acts as a universal standard curve for multiple sets of genes. Accugen is not dependent on amplification efficiencies, and the way to calculate gene efficiencies is more accurate via the window of linearity algorithm. For further information, feel free to contact us.